Hello student, myself Bhanu Solanki. Once again I am here with one more sensational chapter, Molecular Basis of Inheritance. Let's explore the various important areas of the NCERT and interesting facts where there you really need to focus in this chapter. We will discuss all these aspects. In the first section you are watching that there on this paragraph there is the very important thing that you have to keep in your mind. Two types of nucleic acids are found in living systems. While reading a fact or question, you should keep this fact in mind. Two types of nucleic acid, not two types of genetic material. In living systems, DNA is the only genetic material because viruses are neither living nor non-living. So, two types of nucleic acids are present in the living system is a very important aspect. Second thing, RNA, DNA is generally act as a genetic material of the organisms, while RNA has act as a genetic material only in some viruses. Most of the time RNA function as a messenger. It also has some additional role. What this role? It may be adapter, means transfer RNA, or it may be a structural role, means ribosomal RNA. The DNA, a long polymer of deoxyribonucleotides, heteropolymer. It is characterized in terms of the number of nitrogen bases, means base pairs on nucleotide. 5x174 having 5386 nucleotides. Why the unit is written in nucleotide? Because in 5x174 genetic material is single stranded linear DNA. While in lambda phase it is 48,502 base pair because it is double stranded DNA. E. coli again it is 4.6 into 10 to power 6 base pair, means double stranded circular DNA. And then in human being it is 3.3 into 10 to power 9 base pair per genome. It means in a diploid somatic cell of the human being, this value would be 2 into 3.3 into 10 to power 9 base pair. Structure of the polynucleotide chain. The chemical structure of the polynucleotide chain, a nucleotide has the three component: nitrogen base, a pentose sugar, either ribose or deoxyribose, and a phosphate group. There are two types of nitrogen bases as we have discussed in chapter number 9, purines and pyrimidine. Purines include adenine and guanine, pyrimidine includes cytosine, uracil and thymine. Cytosine is the common pyrimidine in both DNA and RNA, while in between uracil and thymine, we must be aware about it that the uracil present in RNA and thymine present in DNA. Nitrogen base get attached on the first carbon of the pentose sugar through N-glycosidic bond, and the resultant structure is known as a nucleoside, such as adenine with the first carbon of the sugar, adenosine, guanosine, cytidine, thymidine, uridine. When a phosphate group is attached at the fifth carbon hydroxyl group of the sugar, then the nucleoside will convert into nucleotide, and the bond that established in between this nitrogen base and sugar is the phosphoester linkage. So one nucleotide have two types of bonds: glycosidic bond in between sugar and nitrogen base, and phosphoester bond in between sugar and phosphoric acid. Now these nucleotides come together and linked through the 3' to 5' phosphodiester linkage and become a polynucleotide. 3' to 5' phosphodiester bond, it means preceding nucleotide contribute its third carbon hydroxyl group and the succeeding nucleotide contribute its fifth carbon hydroxyl group. So the bond is known as 3' to 5' phosphodiester bond. So you are watching that in a polynucleotide chain, in one nucleotide, fifth carbon is free, that is known as 5 prime. And in the another end, 3 carbon hydroxyl group is free, that is known as 3 prime end. So 5 prime and 3 prime ends of a polynucleotide are determined on the basis of carbon which is free, either fifth carbon or third carbon. In RNA, every nucleotide residue has an additional hydroxyl group at the second carbon of the sugar. This is the second carbon, this is the second carbon. On the second carbon, where there, this one is the second carbon. So where there, if OH is present, then it is known as ribose sugar, and if only hydrogen is present, oxygen is missing, then it is called deoxyribose sugar. DNA is an acidic substance. Present in the nucleus was first time identified Friedrich Mischer in 1869. He named it as a nuclein. However, due to the chemical limitation in isolating such a long polymer intact, the elucidation of the structure of DNA remained elusive for a very long period of time. In 1953, James Watson and Francis Crick, based on the X-ray diffraction data produced by Maurice Wilkins and Rosalind Franklin, they proposed a double helical model for the DNA structure. Double helical structure was proposed by Watson and Crick, and for that they used X-ray diffraction data created by the Wilkins and Franklin. Although in this structure, double helical model, one of the important contribution was of Irvin Chargaff, who said that the double-stranded DNA, the ratio of adenine is equals to thymine and guanine is equals to cytosine. Means adenines are equal to thymine. Whatever the number of adenines are there, same would be the number of thymine, and similarly guanines and cytosine number are also constant and it is equal to 1. Means purines are equals to pyrimidine. Means adenines are equals to thymines. Guanines are equals to cytosine. Adenine plus guanine means both of the purine divided by thymine plus cytosine would always be 1. But A plus T divided by G plus C would not be 1. It would be a specific value for a particular species. The base pairing confers a very unique property to the polynucleotide chain. They are said to be complementary to each other, and there if the sequence of the one strand is known, then the sequence of another strand can be predicted out. It was the contribution of complementary pairing. Similarly, if both of the strand of the DNA act as a template for the synthesis of new strand, then two double-stranded DNA that form would be identical to the parental DNA and identical to each other. These are the two properties contributed by the complementary pairing. Some salient features of the DNA are given here, there. It is made up of two polynucleotide chains, where the backbone is constituted by the sugar and phosphate, and the bases are projected inside. Two chains have anti-parallel polarity. Because of the anti-parallel polarity, what is the advantage? That the opposite nitrogen bases come opposite to each other and can take part in complementary pairing. It means one chain has polarity 5' to 3', the other has 3' to 5'. The bases in two strands are paired through the hydrogen bonds, and that is called base pairing. Adenine forms 2 hydrogen bonds with the thymine and guanine forms 3 hydrogen bonds with the cytosine. Because purine is pairing with the pyrimidine, that is the reason that the diameter of the DNA remains constant. So this particular pattern of pairing means purine with pyrimidine generate approximately uniform distance between the two strands of the helix. The two chains are coiled in a right-handed fashion. The pitch means the length of the one complete helix is 3.4 nanometer. While attempting such kind of a questions, be careful, the unit is given in nanometer or Armstrong. In this one pitch helix, there are 10 base pairs are present. Roughly 10 base pairs are present in the each helix. Consequently, the distance between the two base pair, because one helix is 3.4 nanometer, it means in between the two base pair the distance would be 0.34 nanometer. It means 3.4 angstrom. One base pair, then second base pair, then third, fourth, fifth, 10th, and complete circle. It means within this one circle there are 10 base pair. So it means in between the two base pair the angle is a 36 degree. This angle, the plane of the base pairs stacked one above another is the additional, in addition to hydrogen bonds, confers stability to the double helical structure. So number of hydrogen bonds and the plane of the angle between the base pairs that they were stacking one above another are the two regions that provide stability to the DNA. Then this is the scheme given as central dogma of molecular science. DNA to DNA formation called replication, DNA to RNA formation called transcription, and RNA to protein formation called translation. In some viruses, although the flow of information is reverse, means RNA can also form the DNA, and that process called reverse transcription. Because DNA is a long polymer where there two consecutive bases are located at the 0.34 nanometer distance, length of the double helix in the typical mammalian cell is calculated simply by multiplying the total number of base pairs with the distance between the two consecutive base pairs. It means 6.6 into 10 to power 9 base pair if multiplied with the 0.34 nanometer. It means 0.34 into 10 to power minus 9 meter. So it comes around 2.2 meter. A length that is far greater than the dimensions of a typical nucleus, which is approximately 10 to power minus 6 meter. So this much long DNA, how can we accommodate within this extra or this ultra small structure? Then definitely there is a need of a very high degree of packaging. If the length of E. coli is given 1.36 millimeter, can you calculate the number of base pairs present in E. coli? So as we have discussed already, that if the total number of base pair multiplied by 0.34, the total length will come. Similarly, if length is divided by 0.34, the total number of base pair will come. So here there you can calculate. In prokaryotes such as the E. coli, though do not have definite nucleus, we know the word nucleoid, the DNA is not scattered throughout the cell. Instead, DNA being negatively charged is held with some proteins in the region termed as a nucleoid. Means nucleoid is the region where the DNA remains packaged. The DNA in the nucleoid is organized in the large loops, very important point, held by the proteins. But remember, these proteins in the prokaryote are not histones. In eukaryote, this organization is much more complex. There is a set of positively charged basic proteins called histones are involved. This protein doing what? A protein acquiring a charge depending upon the abundance of the amino acid, because histones are rich of lysine and arginine, basic amino acids, means alkaline amino acid. So its overall charge is positive. When negatively charged DNA wrap around the positively charged histones, so overall this structure become neutral, and when it become neutral, its packaging become easier. The negatively charged DNA wrapped around positively charged histone octamer to form a structure called nucleosome. Octamer, two molecules of H2A, two molecules of H2B, two molecules of H3 and two molecules of H4. This is the octamer around which 146 base pair long DNA wrap around it and sealed by histone 1. This is octamer and this is histone 1 for the overlapping purpose. You can see it like by H2A, H2B, H3, H4, and this is our histone 1 for the sealing purpose. In a typical nucleosome, 200 base pair long DNA is accommodated, 146 around it and rest of the 54 to 60 base pairs are present in the linker DNA. Nucleosome constitute the repeating units structure, and in this repeating structure called chromatin, a thread-like structure in which bodies are seen. The nucleosomes in the chromatin are seen, very important, the nucleosomes in the chromatin are seen in the beads on string structure when viewed under the electron microscope. So, chromatin is the beads on string structure in which each nucleosome is the bead, and the rest of the DNA is the linker DNA. Theoretically, how much such beads do you imagine in a mammalian cell? So we know 6.6 into 10 to power 9 base pair divided by 200 base pair would be the number of nucleosome approximately present. The beads on string structure in chromatin is packaged to form the chromatin fibers. It is also called 30 nanometer fibers. They further coiled and condensed at the metaphase stage of the cell division to form the chromosomes. So DNA forming nucleosome, nucleosome forming chromatin, chromatin forming chromosome. This is the hierarchy of the packaging. The packaging of the chromatin at the higher level requires additional proteins, and they are known as non-histone proteins. Non-histone proteins. In a typical nucleus, some region of the chromatin are loosely packed. Because they are loosely packed, so while staining they will stain light colored, and some of the region which are densely packed, they will stain darkly. These lightly stained regions called euchromatin, and darkly stained coiled regions are known as heterochromatin. Euchromatin and heterochromatin both take part in replication. It means replicationally both are active, but from transcription and translation point of view, euchromatin is only the active part. So it is mentioned that the euchromatin is said to be transcriptionally active chromatin, whereas heterochromatin is inactive. Search for the genetic material. Even though the discoveries of the nuclein by the Miescher and the proposition of the principles of inheritance by Mendel were almost at the same time, but the fact that the DNA acts as a genetic material took long to be discovered and proven. In 1926 it started. The question was addressed by the two scientists, I mean to say that the Sutton and Boveri, when suggested that the chromosomes are the basis of inheritance, determine the mechanism for the genetic inheritance had reached to the molecular level. Previous discoveries by the John Mendel, Walter Sutton and Thomas Hunt Morgan and the numerous other scientists had narrowed this search to be search to the chromosome located into the nucleus of the mouse cell. But the question was still remain pending that within the chromosome, what is the chemical responsible for this inheritance. In 1928, Griffith did a series of experiments on Streptococcus pneumoniae bacteria. It is also called Diplococcus pneumoniae. He witnessed the miraculous transformation into the bacteria. During the course of his experiment, a living organism bacteria had changed into the physical form. When Streptococcus pneumoniae, or also called Pneumococcus pneumoniae or Diplococcus pneumoniae bacteria, are grown on a culture medium, some produce smooth shiny colonies while others produce rough colonies. So it means there are two types of strains, S strain for smooth, R strain for the rough. S strains are virulent, they cause death of the mice, while S strains are non-virulent, they can't cause any kinds of a death of the mice. When S strains were injected into the mice, there was the death of mice. When R strains were injected, there was no death of the mice. Third time what he did, he took S strain, but he killed it with the heat and then injected into mice. There was no death, means they were alive. And fourth time, when this S strain heat killed, mixed along with the R strain alive and injected into mice, the mice was die. And from this dead mice, when tissues were dissected, there was the occurrence of S strain of the bacteria alive. It means R strains get transformed into the S strain. Although this transformation, he couldn't explain the mechanism behind it. He concluded that the R strains bacteria had somehow been transformed by the heat-killed S strain bacteria. Some transforming principle, the word that he used, some transforming principle transferred from the heat-killed S bacteria to R strain and convert it into the also smooth bacteria. This heat-killed S had enabled R strain to synthesize a smooth polysaccharide coat and become virulent. This must be due to the transfer of genetic material. However, the biochemical nature of this genetic material he couldn't find out. Then 1933 to 1944, three scientists, Avery, MacLeod and McCarthy, did experiment on the genetic material of the material obtained from the heat-killed S strain, and they assumed that genetic material was thought to be, earlier it was assumed that the genetic material was protein, but after the work of Avery, MacLeod and McCarthy, it get clear that the genetic material is not a protein, instead it is a DNA. What they did? They purified biochemicals, proteins, DNA, RNA, etc. from the heat-killed S cells. Now they allow to grow R strain on the protein, no transformation. On RNA, no transformation. On DNA, transformation. Then they use protease enzyme along with the DNA of the S strain, I mean to say heat-killed S strain, transformation happened. When they use RNAs, then also transformation happened. It means RNAs and protease which were destroying or digesting to the protein and RNA was not affecting to the transformation. But when DNase enzyme was used, as DNA digested, no transformation happened. It confirms that who is the genetic material? DNA is the genetic material. But because of certain limitations and the way this result were published, this result was not thought to be the convincing one. So we need to wait, we had to wait. Here they are asking what's the difference between DNase and DNase. DNA means polynucleotide and DNase means enzyme. It means polypeptide. Unequivocal proof that the DNA is a genetic material come from the experiments of Alfred Hershey and Martha Chase in 1952. They worked on the viruses that infect to the bacteria, means bacteriophages, and we know bacteriophages have genetic material DNA, double stranded DNA. The bacteriophages attach to the bacteria and its genetic material then enters into the bacterial cell. We know that pretty well. The bacterial cells treats with the viral genetic material as it was on and subsequently manufactures more virus particles. They grew some viruses on a medium that contained radioactive phosphorus P35 and some others on the radioactive sulfur medium. Viruses grown on the presence of radioactive phosphorus contained radioactive DNA because phosphorus is the part of DNA, and those which were grown on the radioactive protein, I mean radioactive sulfur, they have radioactive protein because DNA do not have sulfur but proteins may have sulfur. So S35 and P32, these were the two radioactive substances. Those viruses, bacteriophages which were grown on radioactive S35, they have radioactivity in the capsid but genetic material was non-radioactive, while those which were grown on P32, they had genetic material radioactive but protein was not, I mean capsid was not radioactive. First step was infection, second step blending, third step centrifugation. Means allow the bacteriophage to take over the bacteria, then genetic material enter, so we will do blending so that bacteriophage and bacteria become separate, become detached from each other, and by centrifugation they get isolate, they become separate. So when we will do centrifugation, capsid part will constitute to the supernatant and bacterial cell will form the sediment. Here those bacteriophages which were grown on S35, what we found that the no radioactivity detected into the bacterial cells, means into the suspended state, but radioactive was detected into the supernatant. In contrast, when they were grown on P32, radioactivity was noticed into the sediment, but it was not present into the supernatant. It confirms that who went into the bacteria? Phosphorus. P32, it means DNA. Confirmed that the genetic material is DNA. From the foregoing discussion, properties of the genetic material are mentioned here. Remember, RNA is the genetic material only in some viruses like tobacco mosaic virus, QB bacteriophage, while in others RNA perform other dynamic functions like messenger, adapter, structure. Means in the living systems into the cellular system, always genetic material is a DNA. Any chemical that will serve as a genetic material should fulfill following conditions. A molecule that can act as a genetic material must fulfill following criteria. What? It should be able to replicate, yani it should be able to generate its replica. Second, it should be able to chemically and structurally stable. Third, it should provide scope for the slow changes so that evolution can happen. And fourth, it should be able to express itself in form of the Mendelian character, means it should be able to determine a character. These four properties, if any biomolecule is showing, they deserve to be called genetic material. Why, if we examine each requirement one by one, because of the rule of base pairing and complementarity, both nucleic acid, DNA and RNA has ability to replicate. Means from the first point of view, both DNA and RNA can serve as a genetic material. Genetic material should be stable enough, and now as we compare this aspect, so DNA stands far ahead of the RNA. Why? There are some reasons are mentioned. Number one, second carbon of the sugar of ribose, hydroxyl group is present in the RNA is a reactive group and makes this RNA labile and easily degradable. Second thing, RNA has the catalytic activity, so it is very reactive. Therefore, DNA is chemically less reactive and structurally more stable. One more reason is mentioned that uracil is present in RNA and uracil make it more reactive. Presence of the thymine at the place of uracil confers additional stability. So, RNA is structurally less stable. Three reasons are mentioned here. Presence of second carbon hydroxyl group on the sugar. Second, because of the catalytic nature, they are reactive. And third, due to presence of uracil, they are less stable, means more reactive. So from structurally and chemically stable enough point of view, DNA is the genetic material, RNA is not a genetic material. DNA and RNA both are capable to mutate, but RNA being unstable, mutate at the faster rate. Consequently, viruses having RNA genome having the shorter lifespan and mutated fast and evolve faster. So those viruses that have RNA genome and have a shorter lifespan, they will mutate and evolve faster. RNA can directly code for the synthesis of protein, hence can easily be expressed as form of a character. But DNA, however, dependent on the RNA for the synthesis. The protein synthesizing machinery has evolved around the RNA, and the above discussion indicate that both DNA and RNA can act as a genetic material, but DNA being a more stable is preferred storage genetic information. While for transformation of the genetic information, RNA is better because it is not dependent on DNA to express. RNA world. In last NEET, one of the statement based question was asked from this region. RNA was the first genetic material. How we can say? Because the essential life processes such as metabolism, translation, splicing, etc. evolve around RNA. You can see this is one of the assertion reason type of a question. RNA used as a genetic material as well as a catalyst. There are some important biochemical reactions in the living system that are catalyzed by the RNA catalyst and not by the protein. But RNA being a catalyst was reactive and hence unstable. Therefore, DNA has evolved from the RNA with the chemical modification that make it more stable. DNA being a double-stranded and having a complementary strands further resist changes by evolving a process of repair. Because DNA is a double-stranded and it is a complementary pairing, because of this character, one more mechanism evolved into the DNA, that was the mismatch repairing mechanism, that also make it more suitable for to be act as a genetic material. Replication, yani synthesis of DNA from DNA. While proposing to the double helical structure for the DNA, Watson and Crick had immediately proposed a scheme for the replication of DNA. Means semi-conservative nature of DNA replication was first time suggested by Watson and Crick. But they couldn't explain or they couldn't provide any evidences to explain it. The scheme suggested that the two strands would separate and act as a template for the synthesis of the new complementary strand. After the completion of replication, each DNA molecule would have one parental and one newly synthesized strand, means semi-conservative nature. Its semi-conservative nature was experimentally proved by the Matthew Meselson and Franklin Stahl by his experiment in 1958. They grew E. coli on a medium having nitrogen 15, this is heavy isotope, for the several generations, so that the entire DNA replaced by the nitrogen 15, and then after that they do their cesium chloride centrifugation in a cesium chloride density gradient medium. They transferred what they found, that the entire DNA was radioactive, I mean to say heavy active, means nitrogen 15. So the bend that arise in the centrifugation tube was at the bottom, heavy, intermediate, light. After that, what they did, they allowed this E. coli to grow or transfer on the nitrogen 14 containing medium, and after 20 minutes, because we know that in E. coli cells get divided after every 20 minutes. When they did isolation of the E. coli after 20 minutes and do the examination of the DNA, what they found, that the entire DNA was forming an intermediate band. It means it was an hybrid DNA having one strand 15 and one strand 14. After 20 more minutes, means after 40 minutes from the beginning, they collect one more sample, and when they collect one more sample, what they found, that 50% DNA was intermediate and 50% was light. It means nitrogen 14 produce 14 and 15 also produce 15. Here there, when this will act as a double templates, 14 will synthesize 14 and 15 will synthesize 14. So 2 of the DNA would be 14 and 2 of the DNA would be hybrid, means 15 and 14. It confirms that the genetic material is double stranded, is the semi-conservative. If suppose after 60 minutes they will, after 20 minutes they collect sample, 1 bacteria convert into 2, after 40 minutes 2 convert into 4, after 60 minutes 4 convert into 8. This is very common type of a question. So they ask you, if a bacteria already grown in nitrogen 15 medium, when transferred in nitrogen 14 medium, after 20 minutes all bacteria have hybrid DNA. After 40 minutes, what is the ratio of hybrid is to light? Then must remember 2 hybrid, 2 light. It means 1:1. After 60 minutes, if they ask, so you must remember total 8 bacteria will form. Out of them 6 would be nitrogen 14 and 2 would be hybrid. Thus the DNA that was extracted from the culture one generation after the transfer from the 15 to 14, after 20 minutes, all because E. coli divide in every 20 minutes, had a hybrid DNA. DNA extracted from the culture after another generation, means 40 minutes after, was composed of equal amount 1:1 of the hybrid and light DNA. If E. coli was allowed to grow for 80 minutes, it means 16 E. coli will form. So it means 14 DNA would be light, 14, and 2 DNA would be hybrid. Very similar experiment involving use of the radioactive thymidine detects the distribution of newly synthesized DNA into the chromosome. So DNA replication semi-conservative within the chromosome was proved in Visia-Fava, and it was by Taylor and his colleagues. So semi-conservative DNA replication in prokaryote, Messelson and Stahl proved, and semi-conservative DNA replication in eukaryote, then answer is Taylor in Visia-Fava. While you must remember that the Messelson and Stahl work on E. coli. The machinery and the enzymes for the DNA replication. The main enzyme involved is the DNA-dependent DNA polymerase. Means template is DNA and product is also DNA. So we are calling it template DNA-dependent DNA polymerase. Since it uses DNA as a template to catalyze the polymerization of deoxyribonucleotides, so it is called DNA-dependent DNA polymerase. These enzymes are highly efficient enzyme, and as they have to catalyze polymerization of the large number of nucleotides in a very short duration of time. 4.6 into 10 to power 9 base pairs get replicate within a duration of 20 minutes. It means approximately 2000 base pair per second is the rate or the pace of the DNA replication in E. coli. Furthermore, replication is energetically expensive process. Deoxyribonucleotides, very important. Deoxyribonucleoside triphosphate. Deoxyribonucleoside triphosphate means 1 nitrogen base, 1 sugar and 3 phosphate. So deoxyribonucleoside triphosphate, while reading this line, be careful, serve a dual role or dual purpose. What is that? They are acting as a substrate because they are monomers, and they are providing source of energy, because when deoxyribonucleoside triphosphate participate into the polymerization, 2 of its phosphate bond break and release energy that is used for the polymerization purpose. In addition to DNA-dependent DNA polymerase, many additional enzymes like primase, helicase. During DNA replication, opening of the 2 strands is sponsored by helicase enzyme, single-stranded binding protein, ligase, etc. For a long molecule, since the 2 strands of DNA cannot to be separate in their entire length, it means DNA replication will happen in a very short lesion or the length. The replication occurs within a small opening of the DNA helix, and that is referred as the replication fork. DNA-dependent DNA polymerase catalyze polymerization only in 5' to 3' direction. This is very important point. Because it is polymerizing only in 5' to 3' direction, it means on this template 3' to 5', synthesis can proceed continuously in 5' to 3' direction. But in the 5' to 3' template, because synthesis cannot proceed in this manner, it is to be proceed in this manner. So it will proceed in short segments. This DNA, this polynucleotide DNA that arise in a discontinuous manner is known as lagging strand, and this one which synthesizes continuously is known as leading strand. And this nature of DNA application in which one strand synthesizes continuously on 3' to 5' template and another synthesized into the discontinuous manner on 5' to 3' template is known as semi-discontinuous DNA replication. These small fragments are known as Okazaki fragments, and the entire strand is called lagging strand. Afterwards, these strands will join together, small fragments, Okazaki fragments. In NEET, one of the important question was asked that leading strand is growing towards the fork while lagging strand is growing away the fork. Means Okazaki fragments are helping to elongate leading strand or lagging strand? Lagging strand. Towards the fork or away the fork? Away the fork. In E. coli DNA replication start on a particular region, and that particular region is known as origin of replication initiation, in short ORI. Means any DNA which is going to take part into the replication phenomena always must carry origin of replication initiation. Then transcription. Process of the copying genetic material from the strands of the DNA into RNA is known as transcription. Here also the base process of rule of the complementarity govern to this process of transcription. Like replication, only the phenomena what is different, that except adenosine, the component now forms a base pair with the uracil instead of thymine. This is the only difference. Unlike DNA replication, which once set the total DNA of an organism get replicated, transcription happen only in a small segment of the DNA and only in one strand of the DNA. Entire length of the DNA do not transcribe, and only a short segment of the DNA transcribe, and within this short segment only one strand transcribe. It means both of the strands do not transcribe. Here they are giving the reason why. Reason number 1. If both of the strands act as a template during transcription, the RNA molecule with the different sequence will arise. In turn, if the code for the protein, the amino acids in the protein, the amino acids into the protein would be different. Hence, the segment of the DNA would be coding for the two different proteins. It will complicate the process. Second reason they are mentioning here, if the two nucleotide strands produced simultaneously would be complementary to each other, and complementary strands will join together, so they become double-stranded RNA, and double-stranded RNA will not take part in translation, so translation will not happen. So only one strand will take part. This would prevent RNA from being translated. Transcription unit, 3 component, a promoter, structural gene and terminator. You should remember that the addressing of the promoter, template and terminator is carried out on the basis of coding strand. Coding strand is always used as a reference strand. So we can say promoter is located at the 5' upstream to the structure of the coding strand. I repeat again, promoter is located upstream to the structural gene at the 3' of the coding strand, because addressing we have to do with coding strand. Similarly, terminator is located downstream to the structural gene at the 3' of coding strand. If we shift, suppose a terminator I place here and promoter I will place here, so you must remember that this strand will start to act as a coding strand and this will start to act as a template. So here there is a line. Switching the position of terminator, the definition of coding strand and template strand would be reversed. As I said earlier, promoter and terminator flank to the structural gene, very important line. The promoter is said to be located towards the 5' end upstream to the structural gene. The reference is here made with respect to the coding strand. I already said you. In a DNA that provide binding site for the RNA polymerase, and it is the presence of the promoter into the transcription unit that also defined to the template and coding strand. This is the diagram of the transcription unit. Remember, the strand which take part into the transcription is known as a template strand, and the strand which is not taking part is known as coding strand. It means the base sequence of the coding strand and the RNA transcript would be same except uracil and thymine. So 3 prime to 5 prime strand of the DNA will act as a template, and the newly synthesized RNA will also polymerize in 5 prime to 3 prime direction. Yani like DNA, RNA also polymerize in 5 prime to 3 prime direction strictly. It means like DNA dependent DNA polymerase. DNA dependent RNA polymerase also polymerizes ribonucleotides in 5 prime to 3 prime direction. A gene is defined as the functional unit of inheritance. Though there is no ambiguity that the generate the located into the DNA, it is difficult literally to define a gene in terms of what? The portion of the DNA which is capable to produce a polypeptide is known as a cistron, and cistron and gene can be interpreted as a synonyms. If any gene produces only one polypeptide, so we are calling it monocistronic gene, and it is a characteristic of eukaryotes. But if from single messenger RNA arising from the structural gene, more than one polypeptide chains form, so we will call it polycistronic gene. If one messenger RNA, one protein, monocistronic. One messenger RNA, more than one protein, polycistronic. Prokaryotic genes are polycistronic, eukaryotic genes are monocistronic. Again, in eukaryote, the genes are split. Genes in eukaryotes are split. It means it is made up of two parts. What? Expressed sequences called exon. Exons are interrupted by some non-expressive regions called introns. So eukaryotic gene is a split gene in which coding region exons are interrupted by non-coding regions introns, also called intervening sequences. Inheritance of a character, very important line, just keep in memory, is also affected by the promoter and regulatory sequences. Hence, sometimes regulatory sequences can also be defined as a gene, whether they are not taking part into the RNA and protein formation. So every segment of the DNA which give rise to a protein called gene. Any segment of the DNA which give rise to the RNA but not producing to the protein also called gene. And sometimes though, those sequences which are neither producing protein or RNA but can also be defined as a gene if they are controlling to the expression of character. So regulatory sequences, promoters are those segments of the DNA which do not transcribe and translate, but they regulate to the process of transcription. So they are also deserved to be called as a gene. Types of RNA. Here there you must remember, in eukaryotic cell there are three types of RNAs: messenger RNA, ribosomal RNA and transfer RNA. The messenger RNA provide template for the synthesis. Transfer RNA brings amino acid and read genetic code. Ribosomal RNA are structural and catalytic. These are the three important functions. DNA dependent RNA polymerase can catalyze transcriptions all the steps. RNA polymerase bind to the promoter and initiate the transcription. Then it proceed and finally reach to a terminator location. It uses nucleoside triphosphates as a substrate and polymerize template dependent fashion in a complementary rule to the polyribonucleotide. It somehow also facilitates opening of the helix and continue elongation. Unlike DNA replication in transcription, opening of the DNA for transcription is facilitated by RNA polymerase itself. Only a short stage of the RNA remains bound to the enzyme, rest of the part become free. Once the polymerase reaches to the terminator region, the nascent RNA fall off and the process of transcription terminate. RNA polymerase is only capable of catalyzing the process of elongation. But when it is present along with the sigma factor and termination factor, it can initiate and terminate the process of transcription. So what is the question? RNA polymerase catalyze what? Answer, all three steps. initiation, elongation, termination. If they ask, RNA polymerase itself without any additional factor transcribe to what or lead to what part of the transcription, then answer would be elongation. In our NCERT Explorer, this question is given. In bacteria, since messenger RNA does not require any processing to become active RNA, means no post-transcriptional processing, but in eukaryote, transcription is followed by a process of processing, so there is a provision of post-transcriptional processing. In prokaryote, because transcription is not followed by processing, so we can say that the transcription and translation takes place into the same compartment. There is no separation of the cytoplasm and nucleus. The translation can begin much before the messenger RNA is fully transcribed. So you can say that the transcription and translation are coupled in bacteria. In case of bacteria, translation begins before the completion of transcription. Why? Because both happened into the same compartment, cytoplasm. Again, there are additional complexities. At least 3 types of RNA polymerase present into the nucleus. RNA polymerase 1 synthesize ribosomal RNA, 28S, 18S and 5'S. RNA polymerase 3 catalyze synthesis of transfer RNA, 5S ribosomal RNA and small nuclear RNA. While RNA polymerase 2 catalyze synthesis of heterogeneous RNA, or you can also call pre-messenger RNA. Because in eukaryote genes are split, so the RNA that arise is made up of exonic part as well as intronic part, and that is called heterogeneous RNA. Now it will go through the process of post-transcriptional processing, that is called splicing. And this splicing is carried out by a special type of proteins, or you can say ribonucleoproteins called SNRPs, with help of which it happens. Not just only splicing, with help of which capping and talic, cap formation and tail formation also occur here there. Cap get formed at the 5' end of the messenger RNA and tail is formed at the 3' end of the messenger RNA. Tail is made up of 200 to 300 adenylate, means adenine nucleotides. Both cap and tail get formed in a template independent manner. I repeat, cap is the 7-methyl guanosine at the 5' end, and tail is a polyadenylate residue at the 3'. Both cap and tail get formed without any kind of a template, so we are using the word template independent manner. Once the splicing happened, cap formation happened, tail formation happened, this functional RNA is started to be called as messenger RNA. This you are watching introns interrupting to the exon, cutting of the intron and joining of the exons together. This is called splicing. At the 5' end you are watching a cap, at the 3' end there is the tail. So 3 processings we are watching in hnRNA: splicing, capping and tailing. What is the significance of such complexities? The split gene arrangement represent probability and ancient feature of the genome. The presence of introns is the reminiscent of antiquity, and the process of splicing represent to the dominance of RNA world. Because I told you, for the removal of introns there is a need of snRNPs, SNRPs, and they are RNA, means without RNA the transcriptional process cannot proceed into the translation phenomena. Means we are dependent on RNA for our genetic expression. Genetic code. During replication and transcription, nucleic acids were copied by following to the rule of complementarities. The process of translation requires transfer of genetic information from the polymer of nucleotide to a polymer of amino acid. Neither does any complementarity exist between the nucleosides and amino acid, nor could any be drawn theoretically. Therefore, nucleic acids were responsible for the change. There are some examples which suggested that change in the nucleotide sequence lead to the change of amino acid sequence as well. So it means sequence of nucleotide is determining to the sequence of amino acid, but how it happened, because neither nucleotides have any complementarity for the amino acids. To explain it, to support the notion that the changes in nucleic acid were responsible for the changes in amino acids into the protein, lead to the proposition of the genetic code that could direct the sequence of amino acid during the synthesis of protein. Means you can say genetic code is the dictionary with help of which genetic information, coded information of the nucleic acid convert into the sequence of amino acid. Means language of nucleotide convert into language of amino acids. So this process called translation. To determining the biochemical nature of genetic material, the structure of DNA was very exciting, and the proposition of the deciphering of the genetic code were most challenging task. George Gamow, a physicist who argued since there are only 4 types of bases and only 20 types of amino acids are taking part in protein formation, so he suggested that to code for the 20 amino acid, genetic code must be made up of the 3 nucleotides, means triplet nature. So theoretically triplet nature was given by the George Gamow. Dr. Hargobind Khurana was instrumental in synthesizing RNA molecule with the defined combination of the bases, means artificial RNA synthesis. Marshall Nirenberg used cell-free system for protein synthesis so that the genetic material, genetic codes can be deciphered. Severo-Ochoa enzyme, very important, polynucleotide phosphorylase, was also helpful in polymerizing RNA with the defined sequences. It means Severo-Ochoa enzyme was the enzyme that was used for the synthesis of artificial RNAs of the known sequences so that the genetic code can decipher. This is the scheme of genetic codes. Now some features are given. Genetic codes are triplet. 61 codons are coding for the amino acid and 3 are not. So they are known as stop codons or nonsense codon. Some amino acids are coded by more than 1 code, and this nature called degeneracy. Codon is read in messenger RNA in contiguous manner, means no punctuations. Codes are nearly universal, means from bacteria to angiosperm, same code will code for the same amino acid, except some exceptions. Some exceptions are there found into the mitochondrial codons and into the some protozoans. Because of these exceptions, instead of calling universal, we are calling it nearly universal. AUG play a double role. It code for methionine and it act as an initiation codon as well. UAA, UAG and UGA are the stop codons, mean termination codon. If sequence of nucleotides given, so due to that the genetic codes are unambiguous, sequence of amino acid can be found out as given here. But reverse is not possible. If sequence of amino acid is given, so due to degeneracy, because one amino acid can be coded by more than one genetic code, exact nucleotide sequence we cannot find out. From sequence of nucleotide, sequence of amino acids can be predicted, but from the sequence of amino acids, sequence of nucleotides cannot to be predicted. Here a classical example of the point mutation which changes single base pair that lead to the change into the amino acid. If the gene of beta chain of hemoglobin get mutated, a point mutation happen, then instead of the glutamic acid at the 6th position, valine will start to come and the hemoglobin chain become abnormal, and the disease condition is known as sickle cell anemia. Transfer RNA, an adapter molecule. Because amino acids have no structural specialties to read the codon unequivocally, the mechanism here therefore reading to the genetic code was required. So, Crick postulated that the presence of an adapter molecule that would on one hand read to the code and on another hand would bind to the specific amino acid together was realized by the Crick. Transfer RNA, also called soluble RNA earlier, was known before the genetic code was postulated. Transfer RNA has an anticodon loop. This is anticodon loop that has 3 bases complementary to the code and is also as an amino acid acceptor arm. This is the amino acid acceptor arm to which the amino acid bind. Transfer RNA are specific for each amino acid. For initiation, there is another specific transfer RNA that is referred as the initiator transfer RNA. There are no transfer RNAs for the stop codons, so the process of translation will stop. Secondary structure of the transfer RNA has been depicted and that look like a clover leaf. So clover leaf model of the transfer RNA represent to the secondary structure of RNA, while inverted L form RNA represent to the tertiary structure of transfer RNA. Then translation. Process of polymerization of amino acids to form a polypeptide called translation. The order and sequence of amino acids are defined by the sequence of bases on messenger RNA. The amino acids are joined by the bond called peptide bond. Formation of the peptide bond required energy. Therefore, in the first phase itself, important question, in the first phase itself, amino acids are activated in the presence of ATP and linked to their cognate transfer RNA. This process called charging of the transfer RNA or amino acylation of the transfer RNA. If two such charged transfer RNA brought close enough, peptide bond formation will proceed, and this reaction catalyzed by 28S ribosomal RNA of the 80S ribosome and 23S ribosomal RNA of the 70S ribosomes. These 28S and 23S ribosomal RNA are ribozymes and their technical name is a peptidyl transferase. Here there again, ribosomes are made up of ribosomal RNA and 80 different type of proteins. When the small subunit encounter to the messenger RNA, the process of translation of the messenger RNA to protein begins. It is NEET 23 question. Protein synthesis or the translation starts when small subunit encounter to the messenger RNA. Two sites are there, P site and S site. You are watching in a diagram where corresponding cognate transfer RNA is attached. The ribosomes act as a catalyst, 23S in case of bacteria and 28S in case of eukaryote ribozyme. The translation unit in messenger RNA is a sequence of messenger RNA that is flanked by the start codon AUG and the stop codon. And messenger RNA also has some additional sequences called untranslated region UTR. At the 5' end they are present before initiation codon and towards the 3' end they are present after termination codon. For initiation, the ribosome bind to the messenger RNA and the start codon is recognized only by the initiator transfer RNA. The ribosome proceeds to the elongation phase of the protein synthesis. During this stage, complex composed of the amino acid linked to the transfer RNA subsequently bind to the appropriate codon in messenger RNA by forming a complementary base pairing with the transfer RNA anticodon. The ribosome move from the codon to codon along the messenger RNA. Amino acids are added one by one, translated into the polypeptide sequences dictated by the DNA and represented by messenger RNA. This is a very important line. Amino acids are added one by one, translated into polypeptide, dictated by DNA, represented by messenger RNA. Once the stop codon appear on a site, releasing factor will attach here there and lead to the dissociation of this entire structure. At the end, a releasing factor bind to the stop codon and terminating to the translation. Regulation of gene expression. Gene expression refers to a very broad term. Gene expression result into the formation of a polypeptide. It can be regulated at the several levels. In eukaryote, at the transcription level, at the processing level, at the transport of messenger RNA from the nucleus to cytoplasm level, and at the translation level. Because in eukaryote there are various levels of the regulation of the gene expression, so single model can never be proposed to explain the mechanism of gene expression. But in case of prokaryotic cells, as we know, the principal level of gene regulation is the transcription level. So by help of one single unified model, the entire mechanism of the gene expression regulation can be explained. The gene in the cells are expressed and perform particular function on or a set of functions. For example, if enzyme called beta-galactosidase is synthesized by the E. coli, it is used to catalyze the hydrolysis of disaccharide lactose into the galactose and glucose. If bacteria do not have lactose around them to be utilized for the energy source, they would no longer require the synthesis of enzyme beta-galactosidase. Therefore, in simple terms, it is the metabolic or the physiological or the environmental condition that regulate to the expression of genes. The development and the differentiation of an embryo into the adult organisms is also under the result of the coordinated regulation of expression of several genes. In prokaryote, control of the rate of transcription initiation is the predominant site for the controlling to the gene expression. In a transcription unit, activity of the RNA polymerase at a given promoter is in turn regulated by interaction with the accessory proteins. If they affect the ability to recognize the start site, the regulation of gene expression would be here there. These regulatory proteins can bind by showing positive effect as well as negative effect. If they are showing positive effect and proceeding to the transcription, so they are activators. If they prevent interaction of the RNA polymerase, so it means they are repressors. Accessibility of the promoter means promoter will work or not. In prokaryote, DNA is mainly cases regulated by the interaction of the proteins with the sequences termed as the operator. Means if protein interact with the operator, transcription will not happen. If it not interact, transcription will happen. Means the promoter will work or will not work. Who decide? Interaction of protein and operator gene. Leucoperon, Jacob and Monod. Keep in mind, Jacob was genetist, Monod was biochemist. Name of the model, operon model. Examples, leucoperon, tryptophanoperon, etc. etc. Leucoperon consists of one regulatory gene, I gene also called, and three structural genes, ZYA. I gene code for a repressor, Z gene code for beta-galactosidase that lead to the hydrolysis of lactose into the glucose and galactose. Y gene code for the permeasase which increase permeability of the cells to the beta-galactosides, yani lactose, another name of lactose. A gene encode for the transacetylase, LAC-A. Hence, all three genes product into the leucoperon are required for the metabolism of lactose. And in almost other operons also, the genes present into the operon are needed together to perform the same or the related metabolic pathway. Here it is illustrated scheme also. Promoter of I gene, promoter of leucoperon, operator ZYA. Transcription happen, repressor produce, repressor interact with operator, no RNA polymerase can work, no transcription. But if you add lactose into the medium, lactose interact with the repressor and act as an inducer, and its configuration will change, so it cannot interact with operator. So promoter will work, RNA polymerase will attach here and lead to the transcription. Means interaction between repressor and operator, no transcription. No interaction, transcription. Interaction happen or not, who is deciding? Inducer is deciding. Lack operon, if lactose is present, repressor cannot bind with operator. If lactose is not present, repressor will bind. Here one more interesting line that you have to keep in your mind. Expression of the lac operon has to be present in the cell all the time, otherwise the lactose cannot enter into the cell. Very important point. One more. Repressor protein I gene work constitutively, whether the gene is on, lac operon is on or it is off, it will work always. Lactose or allolactose, modified form of it, act as an inducer for it. Regulation of the lac operon by the repressor is referred as the negative regulation because it is preventing to this process. But it is not the only regulation. Lac operon is under the positive regulation as well. So it has both positive and negative, but the impact of the repressor protein is a negative. This is all about the molecular basis of inheritance, very important chapter. 3 to 5 questions can easily be seen in your NEET paper, and now I wish that you are in such a state where there you can focus on the specific exam oriented areas of the NCERT for this chapter. So good luck and all the very best for your exams. Thank you very much.